Okay, yeah, Gilad, by now you have a lot of routine with starting the live stream and everything, I guess, right?
Yeah, it's almost automatic.
Because in the beginning, it wasn't so easy to keep all of these different things straight.
But I still keep making mistakes with the time zones, you know.
Yeah, no, the time zones are a
Time
mess.
they
Right?
change from summer to winter and so on. So
Yeah.
Yeah, I have issues with that even when I'm organizing meetings involving people in the United States, right? not the entire world. I think I'd be totally lost. Hi, Walter. How are you? Haven't seen you in a while.
Hello, Kathy. How nice to see you finally.
Yeah, even in 2 dimensions.
So the next step you think will be 3-dimensional Zoom? Is that what you?
That's right. Obi-Wan Kenobi.
Would be nice.
So we wait a few more minutes so that the numbers will stabilize, and then we will start.
Hi, David. You're muted.
Do you mean even after 2 years of this baloney, I don't remember to unmute myself?
Yeah, I mean, it's stupid enough that these computers haven't evolved a strategy to know when we want to unmute ourselves, right? We still have to do it by hand. It's really
Or Ben,
pedestrian.
even better, a computer that's smart enough to realize that what we're saying is nonsense and to mute us automatically.
Yeah, that might be dangerous.
I think unmute yourself was the term of the year of Merriam-Webster in 2020.
That would make sense, yeah.
Dave just joined us. Hi, Dave.
Oh, that's Eugene. Hello, hello.
Hello, hello everyone. Good to see you all.
So tell me, presuming that COVID actually does have something faster than a power law decay, is this Zoom concept going to continue, do you think? I mean, for this seminar series. Have you thought?
Yeah, we'll have to see.
Yeah, who knows? Maybe this becomes a long-term
Yeah,
thing.
I think it's no way that there will be kind of every month or every 2 weeks in-person conference which bring like 100 people doing protein folding.
That's
So
true.
Zoom will probably be for a while.
Yeah.
I think it's kind of nice to usually your departmental seminars are not right in your field of interest, right? So it's nice to be able once a month to know that you can have a seminar in protein folding. I don't know. Maybe it'll keep going.
No, I think it's, I mean, these kinds of webinars are a nice complementation of existing formats. So in that sense, I do think that these kinds of events will stick around.
Yeah, I agree. Yeah, I mean, they probably would not replace in-person conferences. I hope
No.
so.
No, not replace, just
Yeah,
complement.
yeah.
Okay, Gilad, shall we start?
So yeah, so let's start. So good morning, good evening, good afternoon for everybody. It's a pleasure to have with us today Kathy Royer, or as I learned today, it's actually Catherine Royer. And Kathy is from the Biological Sciences Department in Rensselaer Polytechnic Institute in New York State. A few words about Kathy. She did her PhD actually with the legendary Gregorio Weber in University of Illinois at Urbana. She then postdoc in France, in Paris, on an NIH CNRS fellowship. She came back to the US and worked in Illinois in the laboratory for fluorescence dynamics. And then she became an assistant professor in the University of Wisconsin and went up the rank to an associate professor before she moved back to France to the University of Montpellier. And she was there for quite a few years. And in 2013, she came back to the US to RPI, where she's been ever since. Kathy is currently the chaired Constellation Professor of Biocomputation and Bioinformatics in RPI. Sounds to me like a term from Star Trek or something like that. Constellation Professor. That's really nice. She's very well known for her pioneering studies using fluorescence to probe protein folding, protein interactions, many different aspects of protein dynamics. Kathy has also been very well known for her contributions to studies of proteins with pressure, and she combined her pressure studies together with fluorescence, but also with many other spectroscopic modalities from NMR to X-ray scattering. And she was able, using these studies, to actually discover the mechanism of pressure effects on protein stability and protein structure. And she's made many, many important contributions in this field. She has also made many contributions to, as I said before, to studies of fluorescence of proteins, especially using fluorescence and isotropy, but many other types of fluorescence spectroscopy. And I'm sure she will talk about some of these today. Kathy was recognized for her studies with many awards and prizes. She received an NIH First Award in 1988. She received the Whitaker Foundation Young Investigator Award in 1992. She was an honorary professor and foreign expert of East China Normal University. She recently was the Reinhardt and Dowd Lecturer of Texas A&M University, and many others that I will not read to you. Kathy has been involved for many, many years with the Biophysical Society, and she took many, many different positions in the society, which culminated with her being the Biophysical Society president in 2021 during the COVID year. But still, I believe she managed to enjoy the job of the president. We actually have today with us the president-elect of the society, Tekjip, is somewhere. I saw him before. And she did really great as the president. And I was able to actually see her at work as part of the council of the society. So today, Kathy will tell us about pushing proteins into excited states. And before I give the floor to Kathy, I'd just like to mention that during the talk, I ask you all to mute yourself. At the end of the lecture, we will have a questions and answer session. and you can write in the chat that you have a question, and then I'll ask you one by one to unmute yourself and ask the question. I also want to mention that next month at the 16th of May, we have the next lecture in the series, which will be given by Gabriela Schlau-Cohen from the MIT. So with that, I will stop. And Kathy, please share your screen and you can start the talk.
Okay, well, thanks everyone very much, and to the organizers. It's an honor to be invited to give a lecture in this very prestigious seminar series where I've heard many great talks over the past couple of years, and thanks to you guys for organizing this. It's really been great. So a few weeks back, I heard a great talk by Felix Retort on pulling biomolecules apart using calorimetric force spectroscopy. And today we're going to do just the opposite. I'm going to talk to you about pushing proteins into excited states using high pressure. So pressure actually unfolds proteins, which is somewhat counterintuitive. And it does this because the molar volume of the unfolded state is smaller than the molar volume of the folded state. And a few years back, we set out to try and figure out how this happened in collaboration with Bertrand Garcia Moreno and Angel Garcia and Christian Rumestan. And here I'm showing you some pictures, some of the crystal structures of some cavity-containing variants of this pseudo-wild-type nuclease that were solved in Bertrand's lab by Alfredo Caro. And you can see that there are cavities, new cavities, these green ones, in all of these cavity-containing variants. And here you see fluorescence-detected pressure unfolding profiles for the pseudo-wild-type, and for the I92A cavity mutant, which is this one here, which has a huge cavity in the central core, the OB fold. And just by eye, you can tell that the transitions, these are different denaturing concentrations, but the transitions are much sharper for the cavity-containing variant than for the wild type. And indeed, when these data are fit to a two-state model, we see that the volume change for unfolding is larger for all of the cavity-containing variants with respect to the wild type, pseudo wild type protein. So this is fluorescence detected equilibrium unfolding under pressure. One can also carry out pressure jump experiments. And we did this several years ago. Wisconsin was the first time I did it, actually. It's a while back. And what you can see here is that, again, fluorescence detected, is that the bulk of the pressure effect on wild type nuclease is on the folding rate constant. And we can calculate a sort of Tanford beta volume factor, which is the fractional contribution of the activation volume for folding divided by the total volume change for equilibrium unfolding. And for wild type nucleases, you can imagine looking at this plot, it's a very large number. It's about 90.9% or so. So we did this with these cavity containing variants of nuclease. And what you can see is that compared to the pseudo wild type, all of the cavity containing variants have lower fractional VTSE is what I call this volume beta factor. In fact, the ones that have the cavities really in the deep core of the protein are very, very small numbers. And so these are all of the cavity containing variants in the hydrophobic core. What this tells us is that by putting a cavity inside the hydrophobic core of this protein, you actually don't fold through the same transition state. You fold through a transition state in which probably rather than the hydrophobic core being folded, destabilization by a cavity here causes the transition state, which is of higher energy in the wild type, actually to be of lower energy in these mutants, and involves pretty much the C-terminal part of the protein and not the OB fold domain. So you can get quite a bit of information doing fluorescence coupled with high hydrostatic pressure, but this is going to be the extent of what I'm going to say to you today about fluorescence, even though that's what I was trained in. Science is all about learning new things, and so I turned a few years back with my student Julien Roche, who is now an associate professor at Iowa State University, so it's been a while, to mapping conformational landscapes with high pressure NMR. And we took advantage of the high pressure NMR apparatus that was particularly the high pressure cell, which is a ceramic cell developed by Josh Wand a few years back. And so you can do in this cell all the fancy NMR experiments that you could think of doing at atmospheric pressure. And so we did some of them. And this is just a N15 proton HSQC spectrum of wild type, our pseudo wild type at 1 bar with some guanidine because it's super stable. And you can see that the folded state peaks disappear as we increase pressure and the unfolded states start to appear. You can then plot those just like we do for fluorescence changes and fit them to a 2-state unfolding. And each of these individual residue transitions is a 2-state unfolding event. The protein, the native state is either populated or it's not. So it's a binary thing, but you can see that they do not overlap. And so that means that globally the protein is not unfolding in a 2-state manner. And so having multiple probes, and these are only 4 of the 140 amino acids in nuclease, but we did almost all 140 of them. And so we get information about what's going on across the entire protein structure from doing high pressure NMR. And what Julian decided to do with this, which is really clever, I thought, is to calculate what he calls a fractional contact at any given pressure, at all the pressures, which is just the geometric mean of the fractional intensity of the 2 residues peaks, the amide peaks of 2 residues involved in the native contacts. And so for any pressure, you can calculate this for all the fractional contacts in the protein. And here you see the contacts of nuclease at 400 bar and 800 bar. In black are the native contacts, and blue means folded and red means unfolded. And you can see that the protein starts to unfold here in the C terminus. Whereas the OB fold region, which is here, remains folded. And so that's all pretty good. You can get a good idea of what's going on with the structure, but to visualize it in even greater detail, what Julian did was to construct, based on these fractional contacts at any given pressure, 100 contact lists. And across these 100 contact lists, each individual native contact was present at the fraction, randomly, at the fraction equal to its presence in the fractional contact maps. And then he ran 100 coarse grain simulations using these 100 lists and mapped the configurations back to all atom representations. And so what that gives us is a much more detailed insight into the configurational landscape of this protein under any given conditions. This is the 800 bar that corresponds to this contact map here. You can see you have the folded state at this pressure, and then some intermediates here. And when in these intermediates, the C terminus is starting to get a little weird here. And then finally, this is the unfolded state of the protein. You can also, given the number of residues at any given fraction of native contact, you can, for each pressure that you're interested in, construct a pseudo free energy diagram and see exactly how pressure is affecting the energetics and populations of these states. And so that's, we consider this pretty deep insight into what pressure was doing to these proteins. This is an equilibrium experiment, but we had noticed when we were doing those pressure jump fluorescence experiments that the folding was pretty slow, actually. And so we didn't have to put together what Ad talked to us about, this really super fast pressure jump system in this particular case to examine the kinetics of folding across the entire protein structure. And so again, we have our variants and the pseudo wild type. And here you can see our pressure jump relaxation profiles for 4 different residues. We did all of them, of course, but I'm just showing 4 for clarity in each case. And I'll bring your attention to the axis here, or the time axis. This is 50 minutes. This was the fastest one. But because folding is slow, and because there's a positive activation volume for folding, it gets even slower at high pressure. And so if you look up here at the pseudo wild type or this V66A variant, I mean, it took, for each relaxation profile, it took 24 hours to acquire this. And so we had plenty of time to do two-dimensional NMR after a pressure jump. And so this, we did this for all the residues at multiple pressures for 4 variants of nuclease, which gave us a lot of data to process. And so we would get these residue-specific chevron plots. I'm just showing you 4 of them for one of the proteins. We actually had 15,000 relaxation profiles, I think, to deal with. So a lot of information in these data. Taking the average activation volumes that come from analysis of these plots for these proteins, you can see that, like the fluorescence data, the variants with the cavities in the central OB fold core of the protein showed very low activation VTSE values, and so very non-wild type like transition states. Interestingly, the I92A variant seemed to move toward the excited state, or the folded state, sorry, with increasing guanidine concentration. And if we analyze these VTSE values, we see that the high VTSE values are in the central core of the pseudo wild type. But in the I92A variant at low guanidine concentration, the part of the protein that seems to form the transition state is actually the C-terminal helix, and maybe a little bit of this interaction with the rest of the protein. But as guanidine increases, the part of the protein that is actually structured in the transition state actually moves toward the central core and becomes somewhat more native-like. So again, we have very detailed structural and also energetic information about the folding pathways of this protein. And what you can see is a single site mutation in the core of the protein can really substantially alter how it folds. And I92A actually folds through multiple pathways that are favored or disfavored depending on the conditions under which the experiment is done. So we were interested in trying to understand further the sequence determinants of protein folding cooperativity. And so we listened to our collaborator, Doug Barrick at Hopkins, who's tells everyone all the time, and is right about this, that it's much easier to understand cooperativity in the context of repeat proteins because the interactions are simply between adjacent repeats, and there are no long distance sequence interactions in these kinds of proteins, and it just makes it simpler for our feeble minds to wrap around. And so we applied this approach. We had to, again, add a little urea in this case to get PP32 to unfold. It's a leucine-rich repeat protein. And so you can see, like in the case of nuclease, we lose the intensity of the native state peaks, and the intensity of the unfolded state peaks increases. And again, like nuclease, globally, the unfolding of this protein did not conform to a two-state transition. The residues in the N-terminus lost intensity at pressures that were lower than residues in the middle or in the C-terminus of the protein. Our contact map shows this perhaps more clearly, where the protein starts to unzip from the N-terminus toward the C-terminus. And finally, you can see this in these contact histograms that you can make for repeat proteins. You can see that the N-terminal capping domain is the first to start to fall apart, and the C-terminal capping domain is the last. So it kind of goes this way. And so we ran our simulations and produced a configurational landscape here at 900 bar and 303 degrees Kelvin, and a pseudo free energy diagram. In this case, the computations allowed us to sort of extend to the transition region of this protein. And so what we see here is the first part of the protein to fold is the C-terminus, and this is highly consistent with what Doug found using 5-value analysis. And then as the protein folds, more and more of the structure accretes onto the folded state, and it kind of zips up from the C toward the N-terminus, the last part of the protein to fold here. This is an intermediate. In this position, the last part of the protein to fold is the first repeat and the N-terminal capping domain. So PP32 exhibits this stability gradient from across the protein structure from the N to the C terminus. And so we wondered, how does sequence impact this landscape? And so what we did was to take advantage of all the cavity containing mutants that Doug had made for his 5-value analysis. And so we looked at what happens when you create a cavity in the N-terminus, in the middle of the protein, across the protein in the C-terminus. And in this case, because these are all destabilizing mutants, we didn't have to add any denaturant to these studies. So let me tell you first about the L139A variant. It's in the C-terminal part of the protein. And remember, the C-terminus is where the protein first folds. So this is destabilizing this C-terminal part of the protein. And this protein, unlike the wild type, unfolds completely cooperatively. The amide HSQC, the methyl-bearing side chain HSQC, and even the unfolded state 1D methylprotons all show their transitions concomitantly with a midpoint at about a kilobar here. And the free energies that we get are the same across the sequence, whether they come from the backbone experiments or from the hydrophobic core, and they're relatively narrow distributions and symmetric distributions of these values. We did high pressure small angle X-ray scattering with Richard Gilliland at Chess in Cornell. And we showed that PP32 L139A is a random coil at high pressure. And the Dmax of the paired distribution function almost triples between the folded state and the high pressure state. And the crack key analysis shows that this is unfolded protein random coil rather than a spherical globular structure that you see at low pressure. And so that's L139A. We took a protein that was unfolding non-cooperatively, put in a single site mutation at the very end of the protein on the C-terminus, and made it into a highly cooperatively unfolding protein. What about I7A? And so we do the same thing at the other end of the protein in the N-terminal capping motif. Remember that the N-terminus is the last part of the protein to fold. And so this populates multiple intermediates on unfolding. And so you can see from the yellow, red, green, blue, the transition starts in the N-terminus and moves toward the C-terminus, which is in cyan. That's a C-terminal capping motif. And this, we see the same sort of behavior both in the backbone and the hydrophobic core of the protein. Interestingly, the unfolded state methyl peaks in the 1D spectrum don't start to come up until around 1500 bar. And at that pressure, many of the native state methyl peaks have already lost quite a bit of intensity. And so this protein seems to be populating an invisible intermediate on exchanging with the native state on an intermediate timescale with respect to NMR. So we find, if we analyze these data, that there's a stability gradient from the N to the C-terminus. It's more evident here because we have more residues, and very broad distributions of the free energies of unfolding. When we look at the high pressure sacs, it reveals that this protein, unlike L139A, remains pretty compact. The Dmax only increases to 100 rather than 200 angstroms, so it's a small increase in Dmax. Remains, and the radius of gyration doesn't get very far. This is the random coil value up here. And it doesn't start to do anything until about 1500 bar, when we saw the unfolded state methyl peaks start to come up. So well beyond when we've lost the folded state peaks, both in the backbone and the side chain resonances. And so crack key plots show that the protein, it changes shape, but it remains globular nonetheless, even at 3,500 bar, so pretty high pressure. So this protein, unlike L139A, which is completely unfolded in a very concerted transition, it populates many different molten globule-like intermediates, and we think that the C-terminal part of the protein, because we haven't added urea, remains somewhat structured, and the reason why this RD doesn't go up any farther. So that's the two ends of the protein. What happens if we put a hole in the middle of the protein? And what this L60A substitution does is increase the size of the already existent internal cavity of the protein. Both by amide and methyl bearing side chain HSQC experiments, it looks like a pretty concerted transition at about the same pressures. The methyl unfolded state peaks come up just a little bit later, not exactly on with the loss of the folded state peaks. So it's shifted a little bit. You can see, say, at 1,000 bar here, the transition's almost over in terms of the folded state peaks, but it's only about 2/3 of the way over for the unfolded state peaks. So there's some intermediates in there, but not as obvious as for I7A. We also found that there were intermediate peaks for residues in the C-terminus of the protein that were in slow exchange with the native state. And so we would lose the native state peak intensity, and this intermediate state peak intensity would increase. And so what we think is happening is that the N-terminal part of the protein is unfolding over this range. The C-terminal part of the protein remains intact, but its chemical environment has changed a little bit because of the unfoldedness of the N-terminal part. And so there's an intermediate peak that actually shows that the C-terminus, in absence of urea, remains stable at high pressure. L60A, like I7A, retains globular characteristics. We can see again the radius of gyration is nowhere near the random coil value. Again, it follows the methyl unfolded state methyl peaks in terms of when these changes occur, at what pressure. Again, the Dmax in the pair distribution function doesn't get very large as a function of pressure. The crack key plot shows that the protein becomes elongated, but still, even at high pressure, retains globular character. And some ab initio modeling shows that it's kind of extended, but remains, especially here, it retains globular type character. So cavities in context. So the context of PP32 is that there's the stability gradient across the protein we showed with the wild type. If you insert a destabilizing mutation in the C-terminus of this protein, you abolish this stability gradient, and the protein unfolds apparently cooperatively. Because now the C-terminus is just as unstable as the rest of the protein. In contrast, if you destabilize the N-terminus of the protein with a cavity mutation, you amplify this stability gradient and populate multiple intermediates that are invisible intermediates in terms of NMR. And at high pressure, you still retain structure in the C-terminus, which is very stable, but you populate all these different intermediates along the way in the N-terminus. And then in L60A, you actually have sort of a bipartite destruction of this stability gradient, with the C-terminus remaining stable, and you've just kind of thrown a big rock into the pond here and destabilized the hydrophobic core. And so this part of the protein pretty much unfolds. This part of the protein pretty much does not. And so you have very, very different stability gradients, energetics. You have different intermediates in these different mutations, depending on, they're very similar mutations and sometimes the same mutations, simply in a different context. And so we were curious as to whether the dynamics were really different as well. And so NMR provides all sorts of fantastic ways to probe dynamics. And so what we did was we did high pressure relaxation dispersion measurements and saturation transfer measurements to see if we could probe the dynamics of these proteins. So for I7A, we didn't see any evidence for chemical exchange on the timescale of saturation transfer experiments, which is large, slower than a couple of milliseconds. But we did see evidence for exchange on the CPMG relaxation dispersion timescale. And so what we found was that exchange increased with increasing pressure. So this is 1, 500, and 1200 bar. You can see for this particular residue 15, it's getting larger. The number of residues with an REX value, it's kind of amplitude of exchange here, chemical exchange, increased with increasing pressure. We also found that exchange decreases for I7A from the N to the C terminus of the protein along that enhanced stability gradient. So you can see here at 1200 bar, these are several residues, relaxation profiles for several residues in the N terminus of the protein, and these are for the C terminus of the protein, a bunch of residues. And you can see here that the REX value is sort of inversely correlated to the residue number. And interestingly, also the delta ppm min, which is the minimal distance in frequency between the frequency of resonance of the ground state and the excited state, the invisible excited state, that also decreases with residue number, indicating that the intermediates, the invisible intermediates, may be more disordered in the N-terminal part of the protein than they are in the C-terminal part of the protein, even when they get populated. So I7A exchanges with multiple invisible states on the CPMG timescale, and these increase with pressure but are damped along the stability gradient. With L60A, we didn't see any evidence for exchange on a CPMG timescale, but at lower pressures, remember it was a very cooperative apparent unfolding transition for the amides. And so at lower pressures where the native state amides were pretty populated, we could see evidence for exchange on the CESS timescale. And so here you have a CESS profile for residue 17, for example. This is slower exchange than CPMG, as I noted. And what's nice about CESS is you get the value of the chemical shift for the excited state. And so what we found was the residues that exhibited exchange on the CESS timescale were in the loops of mostly the N terminus, only one out here toward the C terminus. That the chemical shift values of the excited state here in green were much closer to those of predicted for the random coil for these residues than they were for the native state. And so what we suggest is that before that N-terminal part of the protein completely unfolds, it begins to unfold via these loop regions. And that may have something to do with the little differences that we see between the folded state methyls and the unfolded state methyls as well. So up to now, I hope I've convinced you that you can get pretty deep insight into what's going on as your protein unfolds using pressure. Because of the fact that pressure effects are due to cavities, and cavities are heterogeneously distributed in the protein, you tend to populate more intermediates using pressure than you do, for example, with chemical denaturants, which interact sort of in the same way with the entire chain of the protein. And so we've had fun with these model systems, but we know that these excited states are in many cases important to protein function. And we wanted to look at a system in which excited states participate in the functional properties of the protein, and we wanted to map a more functional landscape and not just a conformational landscape. And so I turned to study of ADP ribosylation factors, ARFs, that are small GTPases that control membrane organization in eukaryotic cells. And this is a collaboration with Jacqueline Cherfis in CNRS in France. So Jacqueline's a great friend of mine for many years, and we've been talking about pressure and ARFs for a long, long time, and finally got around to starting to do these experiments. So what I'll tell you about today is unpublished work that sort of hot off the press that we've just gotten on these small GTPases. So let me explain a little bit about these ARFs. So we're interested in ARF1 and ARF6 that perform different functions in the cell and have different specificities. These are signaling proteins. They switch between the GDP bound state and the GTP bound state here. Both ARF6 and ARF1 do the same thing. They do this by rearrangement, pretty substantial rearrangement of the switch regions. So the rearrangements in these little ARFs are larger than, say, in RAS. You may have heard of RAS, and it also is a GTPase, but the rearrangements are larger in these small GTP proteins. And so you can see switch 1 in red is the most obvious thing that happens. It is no longer a part of this beta sheet in the GTP bound form, but rather forms sort of a loop that interacts with the bound GTP. The N-terminal helix, which is myristylated in vivo, is thought to go interact with the membrane, pulling it up and freeing the rest of the protein to go through this transition. And so in the GTP bound state, there's no electron density for the N-terminal helix. And so it no longer caps this region. And so there's a shift in register up of the interswitch, which is the yellow part of the protein, shifts up. And this kind of pulls switch 2 region, which is here in purple, closer to the core of the protein. So it's a pretty substantial shift. And it doesn't occur just on its own. These proteins need associated factors. So to go from GDP to GTP form, they need guanosine nucleotide exchange factors, or GEFs. And to go from GTP to hydrolyze the GTP to get back to the GDP bound form, they need GTPase activating proteins, or GAPs. It doesn't happen all alone. Interestingly, ARF1 and ARF6 recognize different GEFs and GAPs, even though they are very, very homologous in sequence, in particular the parts of the proteins that are thought to interact with the GEFs and GAPs. And moreover, GEFs and GAPs have very low affinities for these ground states. And it's not understood how they do their job with such a low affinity. So a few years ago, Jacqueline and her group solved the structure of ARF6 GTPase that was missing this N-terminal helix. And what they found was that the switch region in one of the proteins in the asymmetric unit was pretty unfolded. And so Jacqueline hypothesized that the switch actually involves considerable unfolding of the switch region. It's not just a shifting around of things, right? It unfolds and then refolds back to the GTP bound form, and that the specificity of the ARFs is due to differential recognition of different excited states that are populated for ARF1 and ARF6 during this GDP-GTP switch. So we set out to try and characterize energetically and structurally, using our high pressure NMR SAX computational approach, what these excited states of ARF might look like. And so thankfully the assignments of ARF1 were already done, so we know what we're looking at. And we see by HSQC N15 proton HSQC, that we lose the intensity in some, but not all of the peaks. But there is no increase in unfolded state methyl peaks at all over this pressure range, even though we lose intensity for a lot of the peaks. The unfolding profiles deviate strongly from two-state behavior. And those that decrease first are the ones in the switch region, so red, yellow, and purple as before. When we fit these data for free energies, we see that the switch region and the N-terminus are globally, except for a few residues, less stable than the rest of the protein. And interestingly, especially the C-terminus has this kind of bimodal distribution of residue-specific apparent stability that you see over here in the distribution of free energies. And looking on the structure, you can see this is what I call the front view, where you can see the switch region here. And that's where most of the low stability is. If you turn it on its side, you can really see that the low stability is in the front half of the protein and the high stability is in the back half of the protein. So this protein is kind of like a sandwich, with a stable front and an unstable back. And you can see this pretty well here at 1600 bar in our contact maps, fractional contact maps, where the N-terminal part of the protein is less folded than the C-terminal part of the protein at 1600 bar. And so again, we ran our simulations. This just shows you that there are pretty significant deviations, excursions that occur during the simulations. And this is at 1400 bar. You can see the pseudo free energy landscape and the RMS distribution, and these high energy states here correspond to these ones with the larger RMSD, of course. And I just want to show you how similar the RMSD plot from the simulations looks to the P of R plots as a function of pressure. There's a small but significant increase in the radius of gyration, but it's certainly not unfolding to a random coil, this protein, a couple of angstroms, but reproducibly so. If we use all the configurations from all the pressure NMR constrained configurations, we see that we get the full folding landscape of ARF1, and you can see how as we go to higher energy states, the switch region starts to unravel here. And so if we explore these higher and higher energy states, we can see that the N-terminus is getting messed up. Here you see the switch region really opening up, and you can imagine that we could bind the GDP and bring it in, or bind the GEF, the guanine nucleotide exchange factor, to switch to the GTP bound state. So, we think that eventually we can provide a plausible pathway for how the ARF1 GTP switch works. One of the things that we're going to do is to go backwards from the GTP bound state, GTP gamma S bound state, and see if we find similar configurations along the trajectory. We also will be looking at the ligand with phosphorus NMR to see what the GTP and GDP are doing during this time at high pressure. In the meantime, we did look at dissociating the ligand completely by adding EGTA, which strips out the magnesium and causes the GDP to dissociate. And this is really interesting in terms of the energy distribution of this protein. It actually completely inverts it, so it destabilizes the backside of the protein. The switch region actually remains the most stable part of the protein. And you can see here, the backside of the protein was either 10 kcals or 4 kcals stable. And here in presence of EDTA, when the ligand is dissociated, it's either 0 or even -4 unstable part of the protein. So you completely invert the sandwich so that now the backside is the weak part of the protein and the front side is the stable part. So we have a lot more work to do. Obviously, we've just started, but exploring the energy landscape and conformational landscape of these small GTPases, but very excited because we think they're going to tell us interesting things about how ligation can modulate and sequence actually can modulate protein function. And of course, that's probably what we're all most interested in. So just to conclude, I hope I've convinced you that pressure perturbation can allow you to populate excited states that are of interest. That the reason this is true is because pressure effects are local, because cavities are local. They're not homogeneously distributed throughout protein sequences. And so that gives you enhanced sampling of these excited states. Coupled with NMR and SACs and computation, we get a pretty detailed vision of what's going on with our protein all across the structure as we unfold it. And so we showed that the context of a mutation, even if it's the same mutation, can really change the conformational energetics and dynamics of a protein. Ligation likewise can profoundly alter protein stability distributions, but excited state populations as well. And so hopefully some of you will think, gee, maybe I ought to try high pressure on my protein. And if you decide to do that, it's not that hard. It's just plumbing, first of all. But second of all, somebody else has done the plumbing for you. And so if you want to do high pressure NMR, you can do this at the New York Structural Biology Center. We do it here in my lab. You're welcome to come here too, but you don't have to mess with me. There's also high pressure NMR available to outside users in New York. And likewise at the Cornell High Energy Synchrotron Source CHESS in Ithaca, there's the high pressure SACs instrument that we use in collaboration with Richard Gilliland. So I'd like to end there just by thanking all my collaborators over the years. Jacqueline's the newest, Bertrand's probably the oldest, Angel and Christian kept me honest in NMR. Julien Roche started all the high pressure NMR. Alfredo did the crystal structures there. And then my group, Martin and Kelly, who've now left, and then JQ here and Siwen here have followed up on the PP32 and ARF proteins, and Tijasri, a new postdoc in the lab, Edgar and Noam, new students here. These three are working now on the ARF project. And of course, I'd like to acknowledge French funding as well as US funding, and thank you for your attention.
Okay, thank you, Kathy, for this really, really beautiful talk and very impressive use of pressure to look at many, many different aspects of protein folding and dynamics. So I guess now we have time for questions, and the first question is by David Nesbitt. So please, David.
Yeah, Catherine, first of all, that's beautiful work, and I'm just totally jealous of sort of the geographic details that having residue by residue pressure effects really provides. It's greatly exceeds what we can do with single molecule studies, that's for sure. But so I have two questions. One is sort of a broad question, which is, I know that you've been doing some work on nucleic acid NMR studies, and I give you the opportunity to talk about whether or not there's a similar sort of richness of information that you get from your nucleic acid studies. But just sort of with this crowd, I was really intrigued by your L60A system, where you had this sort of Jekyll and Hyde, a structured region, and then a pressure-induced unstructured region. And I'm thinking, does this give us some insight into intrinsically disordered protein ideas? And might there be some way in which pressure and voids, that we might be able to understand IDPs a bit better by using that sort of as the contextual clue?
So what pressure does to IDPs depends on whether they have any cavities left. And like alpha-synuclein, I think Ad and Julian did this, other people can speak to this better than me, but doesn't do much under pressure because it's really unfolded random coil type structure, right? There may be residual structure in certain parts of IDPs that can be blasted apart with pressure, and that's a possibility. But recently, Roland Winter in Dortmund has shown something really interesting. And you know, a lot of IDPs form these liquid-liquid phase separated droplet type things. And Julian also has recent work that I just heard about at a meeting last week that shows that pressure actually can perturb these liquid-liquid phase separated droplets. And depending on which protein it is and which temperature and concentration, et cetera, you're using, you can actually, Roland has shown with lysozyme ones that you can start with the droplet, pressure will destabilize it. And then if you continue to higher pressure, you'll actually go back into the droplet state. So there's really a rich place to explore, I think, with pressure and these liquid-liquid phase separated droplets that don't necessarily have to have IDPs in them. They can also have, you know, folded proteins like lysozyme will form them. But yeah, I think that there's a lot to be found there. In terms of the nucleic acids, I noticed that Harish Balasubramanian Harish, who was a postdoc with me and now is working with Roland in Dortmund, is on the call or on the Zoom. And he did a lot of high pressure experiments on RNA in my group when he was here, and is still doing that, I believe, with Roland. And so, you know, double helices don't do that much under pressure, right? But RNA and actually DNA tertiary structures do. So you can blast open G-quads pretty easily. There's a pretty large volume change associated with that. And we've recently got work that isn't published yet, but we're writing up now on a tRNA. And you don't unravel it, but you get shape changes and, you know, in the central core of the tRNA. So interactions between helices seems to be, in this case, pressure dependent, and, you know, the reasons may be different than for proteins. There are probably cavities, but hydration may play a larger role.
Certainly, yeah.
Electrostriction in that case, right? And
what I'm wondering, since your NMR studies residue by residue were so descriptive in terms of whether or not something was a two-state process or it was locally two-state, but then globally multi-state, what do you find for nucleic acids? Or can you tell us what happens for tRNA, for example?
So you have fewer probes. We look at the
Sure.
immunoprotons, right?
Right.
And so we find that there's a little bit of a spread for the tRNA. I'm not making too much of that yet, but it looks to be there's a little bit of a spread. There are regions that are the most highly stable, are the most stable regions seem to unfold or disappear first compared to the other regions of the protein, and that tends to be near where the crystallographic magnesium ion is in this particular tRNA, right? So yeah, there are many things to be probed, and I find myself sort of at the limit of my competence in NMR of RNA, but one does what one can. So,
thank you very much. Very interesting talk.
Thanks.
Okay, next question, Peter Walliness.
Hi, Peter.
Yeah, hello, Kathy. Great talk.
Thanks.
First, a comment, and actually kind of a puzzle. The comment was that, you know, Jerry Jonas, I don't remember exactly when this would have to be, like 1991, 92, used the pressure unfolding on lysozyme, and he was always getting flack from the from the reviewers because he was finding you know, different denaturation plots for different residues. And at that time, I think we thought that was mostly because lysozyme itself is a very complicated folder with having two domains. And so I guess it's a little bit you know, I guess I wonder whether you can fill us in on whether some of the more classical two-state unfolders whose nuclei, whatever, have been figured out by five value analysis of denaturant, whether those have been studied with pressure. That's my first question. And the other one is that we normally think of pressure denaturation as being sort of continuous with cold denaturation, and that they're basically doing very similar kinds of things because we can make a sort of circular phase diagram, in fact. But you didn't seem to talk about that in these systems. Did you look at that?
I haven't looked at it in these systems, but I've looked at it with the CTL9 protein in collaboration with Dan Raleigh, which cold denatures. I mean, it's hard to find a protein that's easily cold denatured. You know, I mean, there are not that many that not that easy to study because water freezes, right? But actually, though, water doesn't freeze at 2 kilobar until you get down to minus 20 degrees. So you can play around with that. But I agree with you that the pressure denaturation and cold denaturation seem to have some similar aspects. The CTL9 protein actually just denatures, and as far as we could tell by SACS, it looked, we didn't, in NMR, it pretty much completely unfolds. And no matter how you denature, temperature, pressure, cold denaturation. But that may not be true for other proteins. And, you know, as we see, all the pressure is going to do is shift to the state with a lower volume. If there's no more volume to be had, it's not going to do anything else, right? And so as soon as the cavities are eliminated, that's all you get. And sometimes that happens when the protein completely unfolds, but sometimes all you have to do is open it up a little bit and the cavity is exposed to solvent and no longer exists, and the rest of the protein, if there's not much volume to be had and it's fairly stable, will remain folded. And so that's why we get more population of these intermediates under pressure. So of the classic two-state folders, I think there have been some studied by pressure, but not so much by high pressure NMR. Right. And so we don't have the information about whether they're really completely two-state or not, or whether you can discern intermediates from the NMR profiles, residue-specific profiles. A lot of work to be done.
Okay, next question, Ben.
Thank you, Kathy. Really wonderful. The combination of pressure and NMRs is a treasure trove. I have a question along the lines of what Peter asked. So you mentioned at the beginning that since denaturants act more uniformly on the protein, that may be the reason why you see less of these stability gradients or deviations from two-state behavior. But I do remember some studies by Victor Munoz who did show on, I think with denaturant, that there were quite some serious deviations from two-state behavior. And I remember this sort of ended up in a big discussion about baselines and NMR experiments. And so I was wondering, what's your take on this, and how do you treat baselines? Is that an issue? Does that sort of pose a complication?
So our signal disappears. What happens in the baseline, so that doesn't have much of a baseline associated with it. What happens is that pressure causes a shift in the chemical shift, and we don't really pay much attention to that. We just follow, you know, the intensity, you know, wherever it is for a particular residue. Whereas denaturants, I think, have a stronger effect on the NMR spectrum of the native state, and so you can have some issues with baselines. I think Victor handled them reasonably well, actually, and that, you know, I think that complexity that he found for that particular protein was real. Just like, you know, I don't think that there's anything wrong with the way that we're doing it, because depending on where the mutation is, we either get completely, you know, concerted transitions or complete chaos, you know, and it's just a single amino acid mutation. So I really, I don't think that that can be.
I agree. I mean, I think in your case with the variants, it's very clear that you have these enormous discrepancies. And I agree. I mean, I also think that what Victor saw at the time was a similar signature. And I
do too.
essentially wanted to get your opinion on this. Yes.
Yeah. No, it was a temperature denaturation, though, wasn't it? Not quantity that I
Okay,
remember,
could be.
that I think about it.
Mhm.
I seem to think it was a temperature denaturation. But anyway, yeah, we don't have much problem with the baselines. It doesn't really move unless we're, the thing completely starts to unfold.
Thanks very much.
Liz Meyering.
Thanks, Kathy. That was just lovely. And
Hi,
I'm full of
Liz.
questions. Yeah, hi. So would you say, I'm just following up on what Ben said, it feels to me like what's happening with your mutations is that you may just be able to observe different parts of the ensemble that's there all the time, right? In your sort of pathways, there's always an ensemble of conformations in the landscape that you may just access or not. Or do you think you're actually changing the pathway?
So for nuclease, we change the folding pathway when we make those cavity mutations. We don't go through the same transition state at all.
Right. I was thinking of your point
for
mutation.
PP32, I would agree. The ensemble is just that the populations of the various configurations in the ensemble are favored or disfavored depending on which mutation you make, right? So to the extent that the L139A mutation just wipes out, even under pressure, the population of any of these intermediates, we just don't see anything, and we look pretty carefully,
Okay.
I mean, they're there, but
yeah, and it's going to depend. I know it's going to depend on the system. So, so my actual other question was, in your pathway for the ARF switch, I thought I saw that something might be happening with the strands. And I wondered if you think, it looks like they're there at the beginning, at the end, and disrupted in between. Maybe they're partially there? I just was kind of struck by how they seem to be changing. Can you comment on that?
Yeah, they're actually in the same position. It's just that they're probably, the angles and whatnot are not exactly perfect for the, pi mold to recognize them as a strand, and so it doesn't draw it that way, right?
I'm just wondering.
But I mean, this part of the strand, obviously is no longer in a beta sheet.
I just wondered, because with our myristal switching and his actophyllin, I wondered for years about whether, that whole beta architecture, the beta trefoil is getting weakened at times and then snapping back. There's some cooperativity there, I think, in this beta strand network, and I wondered if that might be the case here or not, or if it relates to actually the other part, which is your bipartite behavior, and whether you
right. So if you look carefully at where the red means really stable, right? And so, this part of the protein, these two strands are pretty stable. Unfortunately, we don't have much information on the next one, but this last strand at the back of the protein is actually unstable also. It kind of peels off, right? But this core part of the protein here is really super stable in the GDP bound form.
So I think you're saying you don't think that beta sheet is particularly cooperative or whatever. It's
no, it doesn't look like it. No, because these two, of course, in the interswitch, they come off. They're very labile. And then this little guy back here is also the only part of the backside of the protein that's really labile. Is right there. So
very cool. Thank you.
Jose.
Oh, hi, Kathy. That was a wonderful talk.
Hi, Jose.
So it's interesting. Let's go back to a question we asked many times, but the data looks so much better now. Maybe you can answer more.
I'm getting better with age, yeah.
So, like good wine. So pressure affects, like you said, most of its excited states. They are more sort of native-like excited states. They become too disorganized. You lose the cavities and you don't see it. So you see much more the area close to it. What I'm curious is, you make all these comments on cavity. How about water? You didn't comment about water moving in and moving out, having more water cavities and more water separate contacts that tend to change with pressure. How they come on this discussion of cavity? You make everything just comment about cavity. It looks like if you expand a cavity and some water goes in, you may have a different effect. So is that something you observe? How is that story about water moving in and moving out with pressure coming to the picture?
So I'm going to tell you that it depends. So like my CTL9 protein or L139A that I showed you today, that becomes a random coil. And so any cavities in that protein are no longer there. It's not that the water's inside the cavity, you know, the Sachs tells us this is a random coil, right? And so there are no more cavities, water filled or not, right? In this protein at high pressure. But the ARF protein, radius of gyration doesn't really change much, even though there's I didn't talk about it, but a significant volume change. Otherwise, I wouldn't see any difference. You know, it's 100 to 200 mils per mole, depending on the residue that we're talking about, right? But it's not unfolding. The radius of gyration only increases by 2 angstroms. So in that case, I think what's happened is the water has come inside the protein. And, you know, it's one of the classical molten globules, right? Where, you know, we've lost the native state context. They're in exchange, chemical exchange on an intermediate time scale. So we don't see native state resonances, but the protein is not unfolded. Yeah. So, and we don't see any unfolded state peaks from the.
Is that just a very detailed balance of things? Can we predict that, or can we separate that from the data?
I don't know. I don't think we've looked at enough of these systems, you know, because nuclease likewise unfolds completely under pressure, right? There aren't any cavities left in nuclease. It's a random coil. That's why high pressure Sachs is so useful, right? Because you know what the chain is doing. But in the RNAs that David was asking me about, the RNAs don't unfold. But there's still a significant volume change. So there must be water coming into the RNA structure, right, in that case. So I think, like I said, I think it depends on the protein, and I don't know how to predict it. Can you open up a cavity? Maybe you could see, can you open up the structure such that the cavity, the main cavities could be hydrated without disrupting the rest of the protein? Or parts of the protein? Can you retain structure, right? Some structure without disrupting, even though you filled the cavities. And I think that's the question, if you wanted to try and predict that.
Or not totally unfolds. That's interesting.
Yeah. That C-terminus of the PP32 protein doesn't have much volume there, and it's pretty stable. And that's why it remains folded at high pressure unless we put in urea. If we destabilize it with urea, then it'll go, right? But otherwise not.
Okay, Eugene?
Yeah, actually, again, I would join kind of in complimenting you, first of all, for very interesting talk and work. And actually, my question is real direct follow-up to the discussion that you had just a minute ago with Jose. This is about this state at the high pressure, but it's random coil of molten globule. And specifically, I was quite surprised that one mutation can convert from one state to another, from random coil to molten globule. Is there any kind of understanding of that in simulation? Do you have, because you showed quite extensive simulations, can simulations at least kind of hint at this type of effect, whether there is a distribution of cavities which is different? Because my understanding that you need to use all atom simulations for that, it's obvious. But you used all atom simulations, and the question is that...
No, these were coarse-grained.
You mean C alpha?
Yeah, they were C alpha.
They're not a lot. Okay, then it wouldn't be that, it wouldn't work. Yeah, but probably all atom simulations would at least indicate the different distribution of kind of volume, cavities, etc., etc. They might show this difference, because overall it's a really very dramatic effect. And another question that I have is that did you or somebody else in the field look at the time-dependent pressure perturbation. So if you look not at the constant pressure, but at oscillating, with pressure oscillating with time, to get an idea about the elastic losses, real imaginary component, etc., because that will give a pretty good idea about the viscoelastic properties of the denatured protein. Is it possible at all with an MR or not?
Well, oscillating, to do like the frequency response of the pressure. No, I haven't done that. I did the time response with nuclease because it was slow enough that we could do that. We haven't done p-jump, so we did p-jump, right? With nuclease, and we just analyzed it sort of in the classical way for rate constants at atmospheric pressure and for activation volumes for folding and unfolding, right? Just sort of how you would analyze just regular guanidinium hydrochloride kinetics, right? The chevron plots, right? Yeah. So we have pressure chevron plots that we analyze in an analogous way. We didn't look for like viscoelastic responses. But it is possible to do time dependent. It would be very
informative. If it's possible, my feeling is that you can get a treasure trove of information there, in both in time domain and in frequency domain, because it really tells you a lot about viscoelasticity, especially in frequency domain, probably.
Yeah. So there was at the Max Planck in Göttingen many years ago, there was one of these piezoelectric p-jump cells, where they could do that. And the way my system is set up in the NMR in particular, I don't think this would be simple to do. I mean, the time it takes to increase and decrease the pressure in the NMR, I don't think would be amenable to that. But maybe think about it.
So, Kathy, I have a question about the kinetics, the ultra-slow kinetics of the nuclease in the beginning.
Yeah.
I was a bit confused. So what do you think is happening there? Is it still a two-state jump into the unfolded state, or is something else happening there and you get a multiplicity of intermediates on the way?
So for the wild type or pseudo wild type, and also for regular wild type nuclease, we did that one too, also by NMR, but I didn't show it to you. Regular wild type nuclease shows evidence for deviation from two-state behavior in the kinetics, but it's not huge. So what you see is that the OB fold forms, that's what folds at the transition state, and then the C-terminal helix comes and packs against it. So there's an intermediate, an equilibrium intermediate that is you get to after you go over the main barrier, which has the C-terminal helix disordered and the rest of the protein folded, and then the C-terminal helix folds onto the protein. And that's pretty. So it's not two-state, but it's not hugely complex, right? But that I92A variant, it folds through like multiple pathways. One and the pseudo wild type does the same thing as the regular wild type. It just takes longer, but because the barrier is higher. But the I92A variant, which is a single site mutation, the thing folds through multiple different pathways, that are not too distant from each other in transition states in energy. So it's a very complicated barrier for I92A, and which changes with quantity. What?
That basically explains the slow dynamics, you think?
I think mostly the slow dynamics have to do with the stability. So the I92A is not as slow. It's much faster than the pseudo wild type because it's destabilized, right? And so the folded state is destabilized, so it unfolds faster. So the kinetics are somewhat faster for that one, about tenfold. 10-fold, 24 hours, faster than 24 hours is still pretty slow.
Okay, thank you very much. So it looks like there are no further questions. I would like to thank you again for this really wonderful talk. And thank you all for being with us today. And I hope to see you in 1 month in the talk of Gabriela Schlau-Cohen. Thank you and bye-bye.
Well, thanks a lot.
Thank you, Kathy. Thanks,
bye,
everyone.
Bye-bye, everyone.
Very nice, Kathy.
Thanks.